So our next speaker is our most highly watched social media person. His blogs generally hit 500,000 people. A single blog can raise 60,000. Andrew Digby and Deirdre Varco run our Kakapo program. Deirdre is the team leader of the Kakapo and Tākei program. And Andrew is something of a rock star and doc for what he has done for Kakapo, the social media we've been able to put it around, and where he's taking the Kakapo program with Ngāi Tahu in terms of totally new science. So ladies and gentlemen, if we could welcome Andrew Digby from Invercargill.
Sorry, one moment. Tēnā koutou, kua hui mai nei ki te tautoko I ngā taonga o mātou. So welcome to everyone who is gathered here today to meet to help protect and help support our threatened species. And I thought at a crazy and ambitious conference, at a Threatened Species Summit, it was appropriate to talk about this crazy and ambitious idea that we have to protect one of our most threatened species, the Kakapo. And that's our plan to sequence the genomes of all of the Kakapo that are alive today. So Kakapo Conservation has always done crazy and ambitious, and for evidence of that, you have to look no further than this very building. On the third floor, you'll find the infamous or famous Kakapo helmet. And it was a great idea, and like all great ideas, it was simple. So to show you how it works, I don't have to say anything. All I have to do is just show you a short video clip. You have to excuse the lack of sound, but you get the idea. Okay, so unfortunately, that idea did not succeed. And actually next to the Kakapo helmet, you'll also find Chloe. Chloe is a remote-controlled female Kakapo designed for exactly the same purpose as the Kakapo helmet. And like the helmet, Chloe also did not succeed. So both ideas were glorious failures, but I'd argue that bold ideas often fail, and it's more the concept, the intent to innovate, which is more important than the actual success or the failure. And for a case in point, look at Richard Henry. So he was the very first Kakapo ranger in New Zealand, and he failed in his mission to protect Kakapo by translocating them to Resolution Island. But in doing so, he invented conservation translocation. He invented that 50 years before anyone else thought to try it. And his translocations paved the way for another bold idea, an ambitious and a controversial idea by the Kakapo program or the Kakapo team in the 1980s to translocate Kakapo from Rakiura to safe island habitats. Those translocations saved the species. And then since then, the Kakapo team has always had crazy and ambitious ideas. They've always experimented, whether it's through incubation, whether it's through hand-rearing attempts, whether it's through fostering of chicks. The first time that an endangered species the sex ratio has been manipulated through maternal condition by supplementary feeding, the very world first successful artificial insemination attempt in a wild bird. So these are things that have all happened sort of in the past. And of course, the gadgets we have too. We have some pretty amazing gadgets in the Kakapo team. I love gadgets, I love working with them. We have these feeding stations which feed just individual birds. We have proximity sensors. We have proximity sensors at nests. We have smart transmitters as well, which enable us to monitor Kakapo very, very carefully. We can monitor their sex life. In fact, we can even monitor their sex life via satellite. We now have this system whereby when a male booms and a female comes along to mate with him, then the male wanders past a tracking station. That sends a signal via satellite to us, so we know from anywhere in the world which Kakapo have mated with which other Kakapo the night before, whom they've mated with, how long they've mated for, and even the quality of the mating. So we have some cool, nifty gadgets, and this is all the sort of innovation. This comes from Doc's electronics team, who have an amazing relationship with them, and so we're able to do some quite cool things. And so this ambitious project that we have to sequence the genomes is kind of crazy. It's raised a few eyebrows, it's asked a few questions, but it's kind of par for the course for the Kakapo team. But why do we need to do it anyway? And why do we care about Kakapo genetics? Isn't genetics this scary thing which is designed to sort of frighten conservation biologists with all this jargon and technology? I'm actually from an astrophysics background, so I'm very familiar with jargon and acronyms, but yeah, genetics gives astrophysics a run for its money, I think. And I should point out here that I'm not a geneticist, I'm a conservation biologist, so if you want technical genetic detail, don't come to me, go ask a geneticist. Okay, and why do we need to care anyway about Kakapo genetics? And Kakapo in general, isn't the population doing just fine? You might have seen last year we had a record breeding season. We had a 27% increase in the population in just a few months. We've made headlines around the world. The population seems to be doing just fine. And if we look, in fact, sorry, we had lots of warm, fuzzy moments like this with chicks hatching. We actually had 47 of those warm, fuzzy moments last season. And it was a great year. It was an exhausting year, but a great one for us. And if you look at the actual population, over time, you'll see over the last 40 years that it's actually doing pretty well. The decline, then the Kakapo recovery program was started in the mid-90s, and we've had this steady increase since. But if you look closely at this, you'll see that most of the productivity has come from just 3 breeding seasons. And underlying all this is this spectre of genetic diversity, and in particular the spectre of low genetic diversity. Kakapo have got the lowest genetic, or some of the lowest genetic diversity of any species in New Zealand, up there with the black robin. And that comes about for 3 reasons. One is that the Kakapo come from a very small population in total. There was just 63 founders making up the current population. 62 of those came from Rakiura, came from the south of the Tin Range here. And those birds are very, very, have a low genetic diversity. One bird came from Fiordland, here at the confluence of the Gulliver and the Esperance valleys. That was Richard Henry, seen here with Don Merton. Those Fiordland Kakapo are genetically diverse, but we only had 1 bird coming from there. So we have this very small founder pool, and on top of that, we have this added whammy of this weird mating system that Kakapo have. That means that of those founders, only 60% of the males have bred, and of those, only 3 individuals make up more than a third of the chicks which have ever been produced in Kakapo recovery. So this is a big problem. And so genetic diversity is a concern for us. We do manage genetic diversity to govern things and to guide things like nest prioritization, which is something we knew that we started last year, translocation, which Kakapo go to which islands, and also to help us guide our artificial insemination attempts too. So the problem is that we actually have some quite outdated genetic tools. They're good tools, but they're getting outdated. So when the Kakapo genome was sequenced in 2015 by a team at Duke University in the States, that was quite a big deal. That made us sit up a little bit. We're just refining that at the moment. I've just sent over some more samples. They're actually going to turn this into a platinum genome, so the Kakapo will have one of the highest resolution genomes of any bird in the world, and that's a great asset to our program. But for and actually with that genome, I should hasten to add, was just from 1 bird. This is Jane on Anchor Island. That whole sequence came from just 1 individual. And 1 individual is fine if you're an evolutionary biologist like the team at Duke University are. You can just take 1 bird and you can look at things like flightlessness in parrots or brain evolution, for example. But from a conservation point of view, we're interested in genomic variation. We manage Kakapo individually, not as a species. So having individual genomic information would be really important for us. So my crazy and ambitious idea was when 1 was sequenced, I just thought, well, why don't we do all of them? Why don't we all do all 125 that were alive at the time? Why don't we throw in a few dead ones, which are important too, and maybe we can throw in some chicks as they come along as well. So that's the crazy idea, and it's kind of grown from there. But why do we want to do it? What's important about this anyway? Well, first of all, it'll give us a pedigree. It'll give us this great big Kakapo family tree, so we know how all the Kakapo are related to each other. We currently don't know that. It'll also give us a better idea of parentage as well, which we currently struggle with our current low-resolution genetic material. And it'll help us tackle to some of the really big problems facing Kakapo, and probably number 1 of those is fertility. Only a third of Kakapo eggs that are laid turn into birds that fledge. That's a big problem, a big hit in productivity. We get a lot of infertile eggs. We get a lot of embryo deaths. We had a lot of these last year. And there's evidence that inbreeding affects this fertility in females. With the males, we look at sperm quality. This is an amazing sperm sample. We got very excited when we got this one, by the way, last year. This is from Stumpy. Problem is we see a lot of morphological defects. There's evidence also that the sperm quality is related to inbreeding too. So with this genomic information, we'll be able to look at an individual basis why some individuals have less or lower fertility than others. And perhaps the other biggest problem that we have is disease. We have things like erysipelas, beak and feather disease, which are constant worries, but the thing that keeps us all awake at night is this thing called chlorositis or crusty bum. And I apologize because it's just before lunch, and if you're squeamish, you might want to look away because I'm going to show you some graphic photos. This is what a Kakapo cloaca should look like. This is what it can look like if a bird has chlorositis. So it's pretty nasty. It's killed one bird. It probably would have killed many more if we hadn't had treatment for them. And it's escalating. Over the last 15 years now that it's been about, we've seen an increase in the number of cases, especially over the last few years. So it's a big worry for us, but how will genomic information help? Well, let's just have a quick look at something called MHC genes. They're disease resistance genes. And for a population to have resistance against a wide range of these diseases, you need a large diversity of MHC genes. If you look at Kakapo, half of them have exactly the same MHC genes. So that's a big worry. If we also look at inheritance of these genes as well, Richard Henry was the last field in Kakapo. He mated with Flossie, and they had 3 offspring. Now, Richard Henry had these alleles, Flossie had these. All of the offspring got the same alleles that Flossie had, but only one of them, Gulliver, got Richard Henry rare alleles. So Gulliver now is the only Kakapo alive who has those alleles. And people have asked me, why are you doing all the Kakapo? Why not just do the founders? Well, if we'd have just looked at the founders for MHC, we wouldn't know this information. We'd still be treating Gulliver the same as Sinbad, his brother, whereas in fact now we manage them differently. So with this individual genomic information, we can look at many more things. This is just one set of genes. There'll be heaps more like this in the genome that we'll find. Other applications too, so they're the 3 I've just talked about. We'll be able to age Kakapo, which is something we currently can't do. Look at vitamin D, that's a big issue for us at the moment. Population genetics and bioinformatics. How do we deal with these population genomic data? This is a bit of a test case. It's not without its challenges. We've obviously had challenges along the way. One of these is data sharing. We're making the data publicly available to anyone who wants to work on them. We want this to be for the best of Kakapo conservation. We've worked very closely with Ngāi Tahu along this way in how we will deal with this issue of data sharing. And we're coming up with, I think, a good solution for this. When to release the data, and of course the politics as well. It's always the people which is sometimes the tricky thing to deal with in conservation rather than the birds themselves, I'm finding. We've had great support from the Genetic Rescue Foundation who have funded all of this. They crowdfunded it, have raised $45,000 as an initial start, and since then they've raised about in total we've raised about 80,000 New Zealand dollars. We've sequenced now 82 genomes, including the reference. We've got about 56 we want to do to achieve our initial goal. We hope to have that done by the end of the year. So it is a crazy project, it's ambitious, but we believe it's going to transform Kakapo conservation, and yeah, we hope to have it completed soon. So thanks very much for your time. If you've got any questions, feel free to just have a look at our website, follow me on Twitter, or just ask me at lunchtime. Thank you.